All right, so we are going to continue talking about the brain. And just some little fact I always like to read because it just gives you a sense. Even though the brain is just like a few pounds sitting in your head, it's so complicated. I have here a piece of brain the size of a grain of sand contains about 100,000 neurons, 2 million axons, and about 1 billion synapses. So there's a lot of really complicated circuitry and wiring going on in there. Other thing I should mention, people think about the brain kind of in this analogy of a computer, which is up to a point makes some sense, but on some levels also doesn't make sense. So let me talk about the idea of kind of massively parallel processing really briefly. Right, it's when you think about like a computer or something, you do have like your inputs, and then you have like your, this could be like your keyboard or your scanner or whatever, blah, 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 your mouse. You've got the computer, the electronic thing, and then you've got some output, which is like the screen. So it seems kind of like a similar model like we're talking about. But the big difference is what is actually doing the integration, the calculation. Like in a computer, on those little transistors on the chip, you can do crazy amounts of math incredibly quick. You can make, it used to be like megaflops and teraflops. Now it's like petaflops. Like how many billions of floating point mathematical operations can you do per second in the silicon of a computer CPU? In your brain, things are really slow. I mean, we're talking about fractions of microseconds for things to happen in a computer. In your brain, things are happening on the order of milliseconds. It's so slow compared to like what a computer does. But it's only been recently that we have computers that actually can like have a robot that walks without falling on his face. It's like the kind of calculations your brain is able to carry out to do all this complex control of your body and everything is incredibly impressive, but it doesn't do it in the same way like a computer would do it. We have what's called like massively parallel processing. So lots of different parts of the problem are kind of being solved simultaneously, and kind of you converge on the solution. But it's not like in a computer where, even now you often have like, your new CPUs have like multiple cores that are distributing the processing to some degree, but it's still nothing like the way the brain is able to do really complex processing with really kind of slow kind of units. Like we talked about the maximum you can send an action potential down a neuron or an axon is only like 1 kilohertz. Like remember, it's like a 1 millisecond absolute refractory period from one action potential to the next. That means the most you can send down an axon is like 1,000 per second, as opposed to signals going down the wires in your computer, which are going literally billions per second. So it's pretty impressive that your brain does what it does, and it does it through these incredibly kind of complex, nuanced circuits that are kind of distributing the processing rather than just kind of doing it in a more linear fashion. Other things, thinking about the brain. Often I ask, like, how do we know what we know about the brain? I think it's worth talking a little bit about the techniques we use to actually understand, like I'm going to describe, oh, this part of the brain is doing this or blah, blah, blah. Like, how do we get this information? How do we try to understand the brain?
Through
Imaging?
imaging.
So one is imaging. This is more relatively new. Obviously there's been like X-rays. More recently, there's MRI, like magnetic resonance imaging, which you can do anatomically, gives you much more of an ability to image soft tissue. So you can tell like white matter versus gray matter versus CSF and things like that. We'll talk a little bit about things we've learned just about the development and anatomy of the brain through kind of anatomical imaging with MRI. The other thing that MRI does is the stuff I've been doing at Berkeley. You can have functional MRI. This is giving you information about what is the brain doing when in an awake person while they're actually doing some task. Like they're looking at some pictures and thinking about something, and you get to see what parts of their brain are being more metabolically active in the moment, which is, kind of amazing that we have this ability. It's based on what's called the blood oxygen level dependent signal. So it's not actually giving you a readout of neural activity. What it's giving you a readout of is how much oxygen is going into one part of the brain or another at a particular moment, and that is correlated, obviously, with metabolic activity. Parts of the brain that are more active in the moment are going to need more oxygen to deal with their increased metabolic activity. So you'll have an increased bold signal, blood oxygen level dependent signal that you can see with functional MRI. There's some other imaging like PET and stuff I'm not going to go into right now. But this like wasn't even a practical thing when I was in grad school. This is over the last few years, it's been getting more and more practical. Downsides are it's very low resolution. Like you are looking at the brain in little, what we call voxels that are like a few centimeters, maybe 3 millimeters chunk of brain, which is going to include tens of thousands of neurons and stuff all working together. So you don't get like more pinpoint, like what's going on. You get more broad strokes there. And also the time. We have very low spatial resolution with this. Like I said, you can see activity in a kind of relatively broad area of the brain, and also it has very bad time resolution. Like you can see changes over seconds, but not over like fractions of a second, because the signal is like the blood is flowing in, and it's in fact the main change happens like over the next 20 seconds after the metabolic activity increased in the brain. So you both have pretty poor timing resolution, temporal resolution, as well as spatial resolution. So this is good for some questions, but it's very limited in what kinds of things it can look at. What else do we have?
Traditionally, it's kind of a crude way to look at it, but or a crude way to try and understand something. But you take somebody that's had like significant brain injury and see what is no longer, quote unquote, working right.
Yeah, so that's still a really important thing. We call them like lesion studies. Lesion just means tissue damage. It could happen from some injury. Like if you've taken psychology, you've met Phineas Gage, the dude who had like the big pipe went up through his brain, and you could see what parts of the brain were damaged and what were the deficits that showed up in response to that. You can get lesions from, some of them are just genetic things. There's like, we'll talk about a woman who has bilateral amygdala damage. That's just a genetic thing. You can also get them in strokes. This is as good a time as any to talk about strokes. A stroke, its more formal name is a cerebrovascular accident. This is when there is a stoppage of blood flow to part of the brain. It can be because there's some like blockage. You can have some kind of like thrombus that breaks off and blocks the artery and blocking blood flow to parts of the brain. You can have hemorrhagic strokes where there's like a bleeding in the brain, so the blood isn't getting to where it needs to get. If it's only temporary, if it's a short-lasting blockage, you can have what's called a TIA, temporary ischemic attack. People like, whoa, I'm getting dizzy, or like half of my body stops working, but then the blood flow resumes and it kind of goes back to normal. If the blood flow remains blocked, then you start getting more long-term damage and cells start dying and stuff. This is the brain version of a heart attack, right? You can have a TIA in your heart as well, where the coronary arteries have a blockage. Temporarily, it's angina pectoris, crushing pain. And if the blockage continues, then you actually have the cardiac muscle start to die, and that's a more formal heart attack. But so strokes tend to affect one part of the brain, and you can see what kind of deficits. Like we're going to talk about Broca's area for language today. That was discovered 100, 150, 200 years ago. There was a patient who had trouble talking, and when he eventually died, the neurologist kind of dissected his brain and could see where was the damage that was specifically affecting his ability to form words. So that was how they originally found Broca's area, for language, expression of language. But this is still common. People sometimes have a particular accident where a particular part of their brain is damaged, and sometimes there are researchers someplace that are excited to have this particular subject now that they can test and work on. So lesion studies. What are other ways to study the brain?
Would like electroencephalographs be one? Like
Electrode. So this is like an EKG. It's measuring voltages on your skin, but instead of voltages formed by your heart, this is going to be put electrodes on your scalp. And there's going to be some electrical activity that is related to the underlying electrical activity of the brain. This obviously has very low spatial resolution, because whatever is being measured at one of these electrodes is the sum total of all sorts of activity on the brain tissue underneath it, as distorted by all of the scalp and skull and everything. So the good thing, though, about this is it has very good timing resolution, temporal resolution. Like compared to like that fMRI I was talking about, this actually can give you much more detailed information about when things are happening or when responses occur in response to some kind of stimulus. So EEGs, we're going to talk much more about these as we go on. They're used clinically quite a bit, particularly like in sleep studies and things like that, or even like definition of being dead is like when your EEG has no activity anymore that's measurable. There's no electrical activity that you can detect of the brain. So we'll come back to the EEG. What are other ways people have to learn about the brain?
What about like just doing like assessments? I'm thinking with like kids when they're studying them to see if they have ADHD or other learning differences. They have a bunch of assessments and activities they have them do.
So in psychophysics, we'll say psychophysics is not actually poking at the brain, but trying to understand how the brain is processing by just watching responses to stimuli, very controlled stimuli, showing people very explicit, like very specific things on the screen. And can you tell that this is two things versus one thing, or how fast can you respond to it? So psychophysics is this field of just watching how people respond to very specifically created stimuli and using that information to get information to how the brain is processing or what it can do or can't do, or what trips it up. The nice thing about psychophysics is you can just sit somebody in front of a computer with a little response pad. You don't have to like put them in any fancy scanner or cut them open or anything. But you obviously don't get any explicit data on their brain itself. Yeah, other things are actually electrode studies. This obvious, so this actually sticking electrodes on the brain and measuring voltage changes. This obviously is something you have to take the cranium off for, but that happens. Like if you're having brain surgery, sometimes people having brain surgery agree to be the subject of an experiment. There's no pain receptors on the brain proper, so you can put an electrode on the brain and measure what's the electrical activity, or you can stimulate, put a little perturbation and see what happens when you stimulate parts of the brain. Some of the original ones of these, like when we talk about the homunculus for the primary motor and sensory cortices, those were done like in the 50s. What's his name? Penfield. I think those were subjects that had epilepsy. And in fact, we should mention while we talked about strokes, we should talk about epilepsy briefly. Or, seizure disorders. I'll spell it better. What is epilepsy, or what is a seizure?
Is it where blood clots and it can't get to?
So that would be like the stroke, right? The cerebrovascular accident. So that's a very different kind of thing. That is based on part of the brain tissue not getting the oxygen it needs, and so it's stressed or dies. This is about having these hyperexcitable areas of the brain and having like little electrical storms within the brain tissue. So kind of. We talked about how you have a neuron and it's at resting, but it's kind of going up and down. And when it hits its threshold, it sends off an action potential and stimulates the next little part of the circuitry. And you're hoping that everything's working properly and the threshold only gets reached when it's appropriate. In epilepsy, typically there's an area called like a focus. So like here's the brain, and there's some little area that's hyperexcitable and will start just firing off kind of uncontrollably for no reason when it shouldn't. And that tends to then excite the areas around it and create kind of like a little electrical storm in that whole region around there. If it goes across the entire brain, you have what's called a grand mal seizure. Grand mal is like, when people think about like everything going all at once, that's a grand mal seizure. But they don't have to be across the entire brain. You can have them kind of localized. You can have them just like in part of your brain where you like hear sounds. There's ones called petit mal seizures. also called an absence seizure. In a petit mal seizure, people just kind of like go out to lunch. It's like they're there, and all of a sudden it's just like, hello, anybody home? They're like just not responsive. They're not noticing anything. And eventually when they come back, they didn't even know they were gone. But there's no dramatic, like, got a wooden spoon so they don't bite their tongue off. They're kind of just quiet. That's why it's called absence. It's kind of like somebody left the house for a little moment and then came back. These are more common in kids, and often will resolve as the brain gets more fully matured even. But when people have epilepsy, the first step is you try to control it with drugs. Drugs we've talked about, like benzos, things that are going to keep neurons farther away from their thresholds where they're less likely to fire off. But if you have seizures, there's this idea of kindling. Kindling is referring to the fact that if you have these seizures, it causes some damage that makes that part of the brain more likely to have seizures in the future. So that's why you really want to try to control these, because the more they happen, the worse and more frequent they get. One, again, after drugs, they'll actually go in and they'll try to find where is that focus, and they'll actually try to ablate it, it's better to get rid of that little section of the brain that's causing these things than to have them get worse and worse. Because they can actually, it's not just about, in the moment, they can make it harder. Just like I had a student who had a drop a few years ago because her seizure disorder just made it really hard to hold on to memories. Those little electrical storms and stuff just would keep memories from forming as well as they should. So no matter how much she studied, she couldn't. It was, yeah. So it's, you want to try. I mean, I remember seeing when I was in school, we had an interview with somebody who talked about looking at pictures of doing cool stuff with her friends, and all her friends would remember all that really fun day when they took that picture, and she'd look at it and had no recollection of it whatsoever. So the seizures can be worse than just like, you're clenching up or something. So when people are having open brain surgery, trying to get rid of these little focal points where the seizures happen, you can stick electrodes on their brain and see what happens. We're going to talk about Penfield studies where he put them one here and their arm twitched, and put it there and their leg twitched, or put it there and they felt like somebody flicked them on the face. So that's another kind of study. And people do much more invasive electrode studies on non-human animals as well. There's a lot of these studies done on like non-human primates and cats and things like that. One more thing I'll say, just because it's getting more common, is TMA. It's not TMA, that's TMS, transcranial magnetic stimulation. So here your brain is inside your skull. This is a way to actually create electrical stimulations on your brain without having to take your skull off. And it uses a really powerful directed magnetic field. So you have this coil, which you put right on the scalp, but it creates this magnetic field, which then extends to the brain, and if you've taken physics, you know that changing magnetic fields induce electric currents. So by having this very focused, strong magnetic field right around there, you actually induce electric currents in the brain right below there. And you can actually, it's kind of the same thing as if you were actually putting an electrode on their brain and giving it a little pulse. And this is something you can do in normal subjects. So people volunteer for these. And I know people, I've never volunteered because it just seems a little edgy. But other people have done it that I know that, because they also do what they're called like virtual lesions. Like if you pulse it strongly and rapidly for a while, that little part of the brain stops responding for like 5 minutes. So you can like create like quote unquote a virtual lesion and see like, okay, what's not working right anymore now that we've just kind of stunned this part of the brain. And supposedly it all comes back to normal after 15 minutes. But like I said, I've never volunteered for these.
Can I ask you a quick question?
Sure.
Have they tried to use TMS to treat epilepsy, to change the electrical current to stop a storm from happening?
I don't know. I mean, I think they have looked at using it clinically for different things, but I don't think it would be useful for that, because that's more like because you'd have to know exactly where the focal point is and be able to like really just get right into it. And so I as far as I know, it doesn't have clinical use in that.
I have a question as well. Would you consider like autopsies a form of studying the brain, like post-death? Like I know you can't diagnose Alzheimer's until an autopsy is performed.
Right. No, so that's like I was saying, in those lesion studies, that's often when people actually realize what was wrong in the brain. They can observe and document the different deficits, but then, you know, post-mortem, they can then look at the brain in more detail and see what parts of the brain were actually damaged. So yeah, so very,
Okay.
yeah. All right, yeah, so now I'm getting distracted. I like Bonnie's talking and I see like the little Totoro, so all of a sudden Totoro's errors start coming up. All right, so what else? Let's just get into the brain itself now. Let me share. Oh.
Are we gonna go over the warm up today?
Yeah, let's do the warm up first. Let's see, we need to go. All right, tough outermost meninge. Yeah, almost everybody got that, the dura mater. It's kind of like, in fact, if you've been in anatomy, it's basically like a bathing cap that's just over your brain, along with these septa that separate different areas, the two hemispheres and the cerebrum from the cerebellum and stuff. Again, the pia is the one right against the tissue itself, the neural tissue. The arachnoid layer is the one that has that subarachnoid space with the CSF flowing underneath.
Yeah, I was the one who selected the pia matter, and I literally just clicked on the wrong thing on accident.
Okay. So that's, yeah, that's definitely B. Yeah. For this one, it doesn't matter because it's just like 0.2 points. But like, yeah, for the bigger exams, in fact, next Tuesday is your is another mid-unit quiz. So there you want to kind of slow down a bit.
Yes, slow down, exactly.
All right. CSF, ultimately, again, most people got this, is ultimately drained back into venous circulation via the jugular vein. We talked about it. It's being made at the choroid plexuses, then leaving through those little apertures into the subarachnoid space, then from the subarachnoid space to the arachnoid villi into those dural sinuses, and from those dural sinuses into the jugular vein, and then the jugular vein just drains back into your general circulation. So definitely you want to be clear about that whole circulation of CSF. Embryonic forebrain. So it has two main sections. The forebrain becomes the cerebrum, the two cerebral hemispheres that we're going to look at in much more detail today, and as well as the diencephalon. When the diencephalon becomes the thalamus, the hypothalamus, the epithalamus, it actually even becomes like the little optic vesicles and cups that ultimately become your retina, which I didn't mention, but definitely for full credit, you need to know that it's the cerebrum as well as the products of the diencephalon, like the thalamus, hypothalamus. Choose all that are true regarding tracts. They're axons, and they're axons in the CNS. So most people got that. By definition, they're not cell bodies, and they're not glia either. And then myelin, choose all the cells that make myelin sheaths. Again, most people got this as well. It's the Schwann cells and the oligodendrocytes. Schwann cells if you're in the PNS, oligodendrocytes if you're in the CNS. So looks like people were pretty much on track on these things. While we're here, what we're going to do now is really focus on the cerebrum. So again, remember, the brain has the cerebrum, kind of like this big thing on top of all this other stuff. There's lots to the brain, but we're going to focus just on the cerebrum. And we're going to draw it from two different ways. I'll draw it. This would be like what the brain would look like just if you pulled it out of the skull, but didn't cut it apart. This would be just like the sides of the brain. If it's, you look at it, this is that mid-sagittal view. This is if you cracked the two halves of the walnut and looked at the inside of the walnut, like right up and down here. We're also going to be thinking about the brain in this like coronal section where you kind of look at what does the brain look like if you slice it like this. So let me get into this then. So brain's kind of sitting up in your skull like this. If I do this coronal section, what I'll see is kind of these two hemispheres, and then all this stuff in between. And I should make them kind of a little more, little gyri and sulci. These are my cerebral hemispheres. And then I talked about the gray matter of the brain. What did we mean by gray matter?
Tracks.
It's myelinated, I think.
No, so all of that is white matter.
It's deeper nuclei.
In the cortex.
So
The
gray
cell
matter,
bodies.
that's the cell bodies, right? The axonal tracts are myelinated. Those are all white matter. Gray matter is the stuff that's not the interconnected axons. It's the cell bodies that are actually doing like the calculations. And the gray matter is located in this cortex, this thin layer on the surface. So there's cortical gray matter.
You said last class that the cortex is only like 5 to 7 cells thick. Was it? That's mind blowing. I always thought it was thicker than that.
No, it's very thin. And then there's going to be deeper pockets of gray matter, the deeper nuclei. We'll look at the deeper nuclei later. But for right now, what we're going to do is look at the cortical gray matter. So the stuff that's happening right on the surface of the brain. So, in fact, let me see if image. Here, so I'm going to share. Let's see. And this kind of gives you a sense of the gray matter is this thin layer right along the surface. All this other white stuff in here is the axonal tracts that are interconnecting things. Here's some of these deeper nuclei here. Here's like the thalamus. So you can see, but like all of the processing of your vision and your hearing and actually your decision making and all that, that's all happening on this kind of thin layer right at the surface of the brain. So that is the cortex, and that is what we are going to be starting our discussion with. So let's do that. So we're just going to be looking at the cerebral cortex right now. Just everything, when I draw arrows, assume that the brain is not cut open at all. It's just sitting there after you've just yanked it out of somebody's head. Just orient yourself. So here is just looking at one of the hemispheres. If I was like looking at it from the side, there would be two hemispheres, right? Both sides would have like cortex, but we're basically just looking at the cortex, at the surface of this thing, just to kind of give you context. It's worth having the words for the different kind of main regions, the main lobes. We need to know about the central sulcus here. The central sulcus is a little fissure, a little valley that kind of runs down the middle of the brain. It's what separates the difference between what we call the frontal lobe and the parietal lobe. These lobes are named under, by the skull bones they're under. Like the frontal lobe is under your frontal bone. The parietal lobe is underneath the parietal bones. On the back, what do we call the lobe on the back here?
Occipital.
That's the occipital lobe. And then this lobe here, there's this big thing that's called the lateral sulcus that kind of creates a boundary. This is underneath your temporal bone here. This is the temporal lobe. And I'm going to be talking about basic functions that are localized in different lobes. And for the most part, particularly once your brain has matured, there is very kind of specific functions in very specific regions. That doesn't mean that there can't be plasticity or change. I mean, the fact that as you're learning things, obviously your brain is changing because your brain works differently today than it did yesterday. You look at things that you didn't know and now you remember them. So your brain is definitely changing, but there's some kind of core functionality that stays in one region or another region. Also, if there's been damage, there seems to be more of a possibility now that things can like repair or get rewired, or you can this word plasticity is worth knowing. I think I've mentioned it, but I'll mention it again. Plasticity is the term people use for the ability of the brain to kind of rewire and reconfigure. It can just be about kind of, teaching an old dog new tricks, just, creating new reflexes, this or that. It can also be about, repair after damage. Like if things go wrong, can you kind of repair or reconfigure things so you can regain function? We'll talk more about this as we go on. It's limited. It's possible, but it's limited, particularly in adults. In little kids, it's much more, there's much more possibilities. But there is plasticity, and some of it, in fact, I'll talk some of the weird stuff, some of the studies have shown, exercise. Like if you are trying to learn new tasks or try to repair things after some kind of injury, if you do these kind of brain exercises while you're on a stationary bike, it'll more likely have long-term positive effects than if you just sit in front of a computer and do it without the exercise, because there's something about the exercise releasing these neurotrophic growth factors, it seems, that actually helps improve the outcomes and things like that. So, things that affect plasticity aren't always obvious, all right. So let's just talk about some of the main things that are happening in the different lobes. And then we'll go into more details about things. So occipital lobe, again, the back of your brain, this is the primary place where vision is processed. If we go right on the back of the brain, we have what's called the primary visual cortex. Also known as V1 or Brodmann area 17. If you're reading scientific literature where they're studying this part of the brain, sometimes they call it V1 because it's primary visual. Sometimes they call it area 17 because there's this traditional numbering system of brain regions called the Brodmann numbering system, and this happens to be area 17 in the Brodmann numbering system. So just putting that out there, if you ever see something talking about area 17 in the brain, it's just this primary visual cortex. And this is kind of where your kind of more the raw data coming in. We'll talk a little more. Basically, from your retina of your eyes, it goes to the thalamus, but then from the thalamus heads up into the visual cortex. At this level, you can make out some very basic feature things, like maybe the angles of of lines or things like that, but it's very much low level features of what you're looking at. This goes then further up in what we call like visual association areas. So way to think about this is, in fact, I can stop this a second. Book, let me get a book. Here's a book. It's a photo album. It's a rectangle. Actually, I need to turn off this thing. Hold on. Blue, blue, whoop. Not that. Virtual. All right. It's a rectangle. It's slowly becoming a skinnier and skinnier rectangle. And oh my God, now it's almost just like a line. And now it's growing again. Oh my God, what's going on? It's like, it's just rotating. It hasn't changed anything. It's like, but if you think about what is the raw data landing on your retina and going to your brain, this shape and this shape and this shape and this shape and this shape, they're so different. If you were just looking at these as separate pictures, but you don't, you look at it as, okay, you interpret it as a rotating like book. The same thing, like if I took like a penny or a circle, a circle goes from a circle to a line. Or if I have, classic example would be, bump, bump, no, that, bump, bump. If I have something like this, most people will look at this and see like a white triangle blocking, 3 black circles. But that's not what's there. There's no triangle. There's just 3 Pac-Men that are all facing each other, right? But your brain comes up with the most likely scenario, kind of the same way, like if I was, having this and what else? Here's another thing. It's like, you're not thinking like, oh my God, like all of a sudden this isn't a rectangle at all anymore. Now it's got like a big chunk missing out of the middle. And oh my God, it magically reappeared. It's like, no, it was obscured by this other thing. And now I can see it again because it's not blocked anymore. But that's not what's on your eyeball. On your eyeball is just the raw image. Like this thing now does no longer, this gold thing now looks like a thing with a notch in it. And now it doesn't have a notch anymore. So this idea of integration, you look around and you don't just see lines and colors and splotches, you see objects, and you see objects relating to each other or obscuring or doing things. So that's when I talk about that integration. It's kind of this interpreting what this data is versus just what is the raw data there, if that makes sense. And we're going to see that same idea. There's going to be auditory association areas where you don't just hear blips and blops and frequencies and things. You hear my voice, or you hear like some piano coming through leaked if it's not muted, or, and it's like, I'm not like trying to like, oh my God. And even I can hear my voice and the piano at the same time and know they're two different things and make sense of what it is. But on your eardrum, it's just one summation of all of the different vibrations all happening at once. And somehow your brain teases it all apart and actually kind of makes sense, creates these individual auditory objects, which it can then look at individually. Same thing with touch. We're going to see sensory, somatosensory association areas. If I'm holding on to something, I don't just feel pressure and temperature on different digits of my hand. It's like, oh, it's round and smooth. And, that's this, somatosensory association. I'm kind of integrating that raw data into a more sophisticated interpretation of what it all means. So does that make sense? Because the basic information coming in is not telling you that you're looking at a particular object. You have to figure that out. All right. Blah, blah, blah. All right, so we have primary visual cortex. We have our little visual association areas as you get further up. I should mention one of the interesting things, another way that brain is very different from a computer. Actually, so let me talk about this idea of mapping briefly. So mapping is kind of the correspondence of the physical world onto the areas of the cortex where it's being processed. So if I am looking out at the world out here, comes in my optic nerve, my thalamus, it gets initially processed on neurons here in the visual cortex. There's actually this kind of pretty predictable correspondence between the physical world that I'm looking at and how it's being represented in the cortex. There are these experiments that were done a while ago where they're kind of sad. I'm gonna like make it not even look real so we don't have to think about it being a real guy. They took a cat and basically kind of wire its eyes open, have it stare at a grid, and then sacrifice this, and then inject them with radioactive glucose. So radioactive glucose, the neurons that are being particularly active are going to need more glucose because they're metabolically active. So the cells that are really active as this cat is watching this grid are going to take up more of the radioactive glucose. So then you can sacrifice the cat and you can spread out its visual cortex on a piece of photographic film, and the parts of the cortex, the brain that took up more radioactive glucose, will expose the film. And you see that there is kind of this grid representation, like the representation of the visual world mapped out to a representation on the cortex. Like if I am looking at my Zoom screen, I see like, Ethan and Irene are really close to each other in their little boxes, at least in my vision, and they are going to be represented, nearby in my visual cortex because they're close in real life. Whereas I look at Ethan and Candace, and they're on opposite sides of the screen, they're going to be represented farther away from each other on the on my cortex. We're going to see this kind of mapping happening all over the place as well. We're going to see how sound, different frequencies are mapped in a predictable way along the auditory cortex. There we're going to have what's called like tonotopy, mapping of tones. We're going to see somatotopy, how the body is mapped out along the somatosensory cortex. So it's kind of how the world is represented in the cortex in this kind of regular way. So that being said, exactly how the world is represented in the cortex can actually change depending on how you're paying attention to things. So, we're going to talk about the prefrontal areas. This is kind of the executive areas up here. Executive functions, again, what we call the prefrontal area. This is where you're kind of making decisions and things. So most people, again, take the analogy of the computer too far and just think about, oh, the data is coming in and it goes up here and then you decide what to do and then you have some response. But it turns out that there are a lot of neuronal tracts that go the opposite way. In fact, there are more neuronal tracts that actually go from these prefrontal areas to these lower areas that are actually going from the lower areas up to the higher areas of the brain. Like you can show, like one of the things actually that the guy I work with at Berkeley, he showed that if you are using covert attention, if you are, so covert attention is I am looking at one thing, but I'm paying attention to something off to the side. Again, this is so much easier to do in person. Let's see, what, stop. Okay, I wanna, so trying to think, like, let's say I'm, again, normally be like, I'm really worried about what's going on with this little like pen over here, but I don't want this pen to know, or actually Elsa, I got Elsa. Here's Elsa. Elsa is hanging here and I'm worried that Elsa is up to no good. She's gonna like freeze the kingdom and cause havoc or something. But I don't want her to know I'm worried about this. So I'm just like looking at the cat. But I'm actually putting my attention on what's Elsa doing over here, even though I'm looking at the cat in front of me. That's why we call it covert attention. She doesn't know that I'm paying attention to her because I'm not looking at her, I'm looking at something else, but I'm actually putting my attention over to this part of my visual field where she's doing her ice magic or whatever. So they've shown that the way that the visual world is represented in your visual cortex when you are doing this covert attention actually changes, that you will actually kind of reallocate more of your cortex to process that part of the visual space where like Elsa was. So I have more brain power to like kind of follow and see what's going on there. So your attention actually changes kind of how your very first levels of visual processing actually work. So it's not just, oh, here's all the raw data coming up and then percolating to get sifted through. Like how you are actually holding your attention actually changes the configuration of kind of the primary visual cortex that is taking in the raw data. So I think that's always interesting to remember. People always often talk about like, the world you see is affected by the world you're looking for. So there's a lot of truth to that on many levels, but even on this very explicit level of how it's the visual world is being represented in your primary visual cortex. Okay. All right, so there we've done the occipital lobe, which is pretty much visual. And there are visual areas that extend up into the parietal lobe and beyond as well. So it's not just there, but that's the primary place. You know, and if you have a stroke that obliterates part of your visual cortex here, it is going to mean that you don't have any information anymore about that piece of the visual world. I had a student who was in a horrible car accident where she snapped her femur and there was fat, you know, if you remember from anatomy, the marrow is mainly fat. So all this fat ended up getting loose into her bloodstream, and one of those fat globs ended up clogging up one of the arteries that supplies part of her visual cortex. And she lost part of her visual cortex from that stroke, from that fat glob, from breaking her femur in the car accident. And she eventually came back and took the class again. But we could play around. Like there was just parts of the visual field, like that just didn't exist. Like, she could see everything. It would be like a hole, but a hole you didn't even know is a hole. It's just like part of the visual field just doesn't exist because no information was coming in to get processed, to get sent further on.
I have a question about that.
Sure.
I've certainly never heard of that happening. Do you think, like at some point, was that something that possibly somebody should have tested for? And because with the stroke, you have, the golden hour and all that. I know they had a lot to tend to in her case, but is there ever, I don't know.
So that I can say, I don't know. I can tell you the story she related, but in my training is not clinical in that way. So I do know, blocking that kind of a clot would be, because a lot of that golden hour is to give them clot buster drugs, which is going to break down fibrin, but this was not fibrin. This was embolus made out of like a fat glob. So I'm not even sure what you could have done, even if you knew.
I'm also curious. I broke my femur a long time ago, a really bad break, and so far I feel like my vision is fine. But that's really alarming to hear.
It was in the moment. It was in the moment when the bone was open. It's not something that's going to happen later on.
Oh, I'm not worried about it now. No. Wow, that's crazy.
Yeah. So there are. We'll talk more about. You can actually. We'll see that when we do our visual thing next week. We'll see the blind spot where there's parts of your visual field that you don't see that you just make up. If there's a large enough area that's missing, you can actually especially on the retina, you can actually start making up weird cartoons and stuff. It's kind of bizarre. But blah, blah, blah. Let's continue on. In the temporal lobe, it's got a variety of things. Let's focus on the primary auditory cortex. So primary auditory cortex, this is where your auditory information is coming from. In fact, I want to get the. So it's lower. Okay, so there's tonotopy here. Or the mapping again. Instead of mapping of the visual world, it's mapping of frequency. So like lower frequencies, like are going to be processed by the neurons on this part. And as they go, it's going to be processed by neurons. So again, this mapping again. But instead of, the representing a visual space on the cortex, it's the representation of frequency on the cortex. And again, like I said, there's going to be association areas for all these senses. Right? You don't just hear random frequencies and stuff. You hear auditory objects. You can figure out my voice versus like somebody else calling you in the background versus your dog or your cat purring or the refrigerator going and humming, and it's all happening at once. And we'll talk more about that when we do the auditory system, but it's kind of amazing when you think about sound. Sound is all the air vibration coming in. You have an eardrum that vibrates, and somehow you're able to like tease apart that big just mishmash of everything and turn it into, a sensible interpretation of what around you is probably creating these vibrations that we're perceiving as sound. So we'll talk much more about the auditory system. Let's looking at the time, let's continue here. Right on the posterior of this, so the central sulcus is this groove that's kind of going down here. This was the central sulcus. Right behind the central sulcus in the parietal lobe here, we have what's called the post-central gyrus. The post-central gyrus is called also the primary somatosensory cortex. So somatosensory, these are like the general senses, like the touch and stuff on your skin. So we're going to be looking at a bit in lab today. So when you are getting this touch on you or pressure or things like that, that's getting processed here in this post-central gyrus. And again, there's a mapping of your body onto this post-central gyrus, where one finger and the next finger, and then your arm, and then your leg. In fact, there's this word people use, the homunculus. Means little man. It's like if you take a picture of a person and you kind of draw how those parts of the body are represented on the cortex, you get a nice little map. And I'll show you. Blah, 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 blah. The homunculus, like you can, this is supposed to be, so can I open? Right, this is supposed to be cortex, this kind of, the dark brown. This is supposed to be like what parts of the cortex are representing different parts of the body. Like in these experiments in the 50s that Penfield did, he had these people with the top of their brain off, with top of their skull off for these epilepsy surgeries. And he'd go in with a gentle electrical stimulation and he could map this out. He could, oh, I feel a little twitch on my arm. I feel a twitch on my neck. I feel a twitch on my hip. I feel a twitch on my ring finger, my middle finger, my thumb, depending on where he did the little electrical stimulation. So you could just find this mapping, this correspondence between the different parts of the body. Sometimes they call this somatotopy, kind of the mapping of the body onto the cortex. They often call this the homunculus, the little man, because, and if you take a look, it is not proportional to the size of the body. What parts of the body get the lion's share of the representation on the cortex?
The hand.
The hand, and it gets cut off on this picture, but the face and the tongue as well. Here, this is a better one. This is, and we'll see this the motor homunculus is the similar kind of thing. Both looking at the somatosensory cortex as well as the motor cortex. The face and the tongue and everything, as well as the hand, each get like about a third of the real estate, which makes sense. Your hands have incredibly fine sensory discrimination as well as motor control. Your face is also, that's where expression, how you talk, you have to have incredible motor control and all that to make all of this stuff work. So, you know, your hands and your face and mouth and all that each get like about a third, and then the rest of your body gets the rest. So it's very unfair in terms of the mapping out. You know, there is some plasticity. They've actually done, let me get a little drawer thing. They've done these, they're kind of horrible experiments where they'll take a monkey and they'll train it to do different tasks, and then they'll look at, you know, map out the details of how the hand is represented on the cortex here. Here's one finger, the next finger, the next finger, the next finger, and then they'll amputate the fingers, and then they'll have the thing, the monkey keep learning the task and use its brain with the limited limbs it's got, digits it's got left, and they'll find that there is some plasticity, like parts of the cortex will kind of take over and remap. Although this does seem like, if people have heard of phantom limb pain, sometimes people will lose an arm, but then still feel like there's an arm there, or sometimes there'll even be pain in this hand and they can't make it stop. And there's nothing you can give it for them because there is no hand actually that's making the pain. What they think is happening is if you lose a limb, you know, that part of the cortex that represented that limb is still there. Or just be on the sensory side makes more sense. And if there is electrical activity in the neighboring areas that happens to create some response in this part of the brain that used to represent the part of the body that's missing, you can still have a subjective experience of something happening in that part of your body, even though that part of your body doesn't exist anymore. Because ultimately, you know, your whole experience of the world exists in your brain. It's the matrix, right? You know, you have sensory information coming in from receptors in your body that ultimately trigger activity in your cortex, and that activity in your cortex is what you actually perceive as somebody touching you. But if you stimulate activity in that part of the cortex, even if nobody's touching you, you'll still think someone's touching you. Right? That's what I was saying. That guy Penfield would just take electrodes and stimulate parts of the cortex without touching the body, but people would still have the experience of somebody touching their body, because that's typically what happens as a result of someone touching your body. There's signals from the receptors up the axons, the nerves, and then create activity in the cortex. So blah, 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 blah. So back to where we were. So we had our post-central gyrus. And again, I want to make sure, you know, it's the back. Which is our primary somatosensory cortex. And it's in this parietal, it's called the parietal lobe. Everything behind this central sulcus is the parietal lobe, except for the occipital lobe, obviously. and in the front is the frontal lobe. There's association areas as well. Like I said, I don't feel this as just different touch and pressure on the digits of my hand. I feel round and smooth and whatever, right? So you can take the raw sensory, somatosensory touch data, and you interpret that as something more useful for you in your life. The precentral gyrus. This is the ridge that's right in front of the central sulcus. This is going to be what I call my primary motor cortex. This is going to have basically pyramidal neurons that go down and leave the brain and operate your skeletal muscles. Again, Penfield, in these experiments, he would take an electrode and go along different regions on this primary motor cortex and could make different parts of the body twitch. Twitch a finger, twitch the other finger, twitch the leg, twitch the toe. There's some evidence that what's going on in the primary motor cortex is a little more complicated than just purely direct activation of one or another piece of the body, but that's a good first approximation. This is kind of where you are operating your voluntary muscles. The axons that come out of here, this gives rise to the pyramidal tracts. If you remember from anatomy, there's the pyramids going down the brainstem. Those are the big axons that are going to operate all of your muscles. We'll also be talking about some extrapyramidal motor pathways a little later, things that aren't coming directly from the primary motor cortex. So it's worth knowing that these pyramidal tracts, they get their name from the pyramidal neurons that are sending out the axons. When you see this word pyramidal tracts, it just means tract, bunch of axons going in the CNS, and they're carrying out these motor signals from the primary motor cortex. Obviously, you don't think about your motion in terms of operating some little piece of your musculoskeletal system. So we're going to have motor association, sometimes called premotor. The basic idea here is kind of the motor plan. Like if again, this is another, it's way easier to do if, there's also, again, it's like, let's say I want to see, someone says, oh, like, touch Elsa on the nose. So I go in, it's like, ooh, I touched her on the nose. It's like, that was super complicated though. I had to like kind of bring my arm up. It's like, okay, wait a second, what muscle is that? Oh, I'm probably using like some deltoid. I'm doing this medial rotation. So there's maybe infra or teres major. And then I'm doing this. I'm doing all these muscles to move my hand. I don't think about that. I just think, move my arm and hand to touch this little piece of paper. You have incredibly complex planning that has to happen for you to do something simple, or if you get pulled over and it's like, touch your nose or whatever. It's not simple, but it's simple for you as like when you're not thinking about it, but it's not simple for your brain. Your brain has to do all sorts of crazy, complicated, like I did my original undergrad work in engineering and spent a lot of labs designing control circuits for robotic arms and things like that. How do you take a physical, a mechanical system that has moments of inertia and has little actuators, and you want it to do something, and you want it to start and go fast, but then stop where it's supposed to stop and not overshoot and maximize accuracy and speed. And it's not simple, especially when you have this big complicated like mechanical system with all these different movable joints and everything. And your brain does a great job of making it do what you want it to do in general. And we'll talk about even like moving your eyes. If I'm looking here or I'm looking there or looking there, that's a lot of planning. We're going to talk about parts of the brain that can calculate where do I want my eyes to be looking, and how do I have to frickin' accelerate my eyeballs using my 6 extrinsic eye muscles for each eyeball and pull them and slide them and land my gaze right on this new part of visual space, bring them there as fast as possible, but not overshoot and just land smoothly. It's not trivial for your brain circuits, or I should say the calculations aren't trivial, but for you, it's just all automatic. There's so partly I like bringing this stuff up just so you don't take for granted what your brain is doing. And we'll get to Broca's area in a few moments. The fact that I'm talking to you is crazy, right? I just have ideas in my head, and there is such complicated manipulation of my tongue and my lips and my mouth and my to make words happen. It's crazy, but I don't think about it. I just like have a thought and the words vomit out of my mouth. It's that's we're going to see Broca's area is related to that motor association area. It's located kind of down at the base of that. So speech production is a specialized version of this, motor association, because it's what do you have to do to operate your muscles in order to create speech. Okay. All right, so we have occipital with kind of visual, somatosensory here. We had auditory here in the temporal. We have motor. Before we take a break, I'm going to add one more thing in before, this prefrontal area. So it's called prefrontal because it's kind of in the front of the frontal. It's still frontal, but a lot of frontal is motor, as we saw here. So prefrontal, this is the part that we talk about being executive functioning. Again, the executive of a business is the one who makes the choices. We're planning for the future. Impulse control. Personality. Decisions. If I can spell decisions. When you have lesions that damage the motor cortex, what are your kind of deficits going to be?
Movement.
Movement. You're going to be like paralyzed or some problem with your movement. If you have deficits with your somatosensory cortex, you're going to lack feeling. If you have deficits in your visual cortex, you're going to have problems with your visual processing. When you have lesions that affect your prefrontal area, sometimes the deficits aren't so obvious at first, but they can be very severe. Like, the classic thing with Phineas Gage, which I mentioned earlier, who's like kind of the poster child for prefrontal lesions. He had this steel pipe go through his brain. It didn't hit anything that was critical to stay alive, but it really changed his personality. He went from supposedly being a nice guy to being an asshole. Impulse control, in part of my schooling, we met people with different kinds of problems. I met one guy with prefrontal lesion who, more from talking to the people who work with him, like if he just saw a woman he found attractive, he would like reach out and try to touch her breasts. It was like, there was not that extra part of like, okay, what do I want to do versus what should I really be doing here and what's appropriate. So impulse control. I mean, one of the fascinating things that has come out of the brain imaging, like with the MRI that lets you image soft tissue, like myelinated axons versus neurons, is they found that as the brain develops, this is the place that seems to develop last. Like they will look at the brain. And again, when you're born, you've got like twice as many neurons as you're going to have as an adult. And as the circuits wire up, you get more and more myelinated axons in the mature circuits. And what they find as they look at the brain from being like a baby to an adult, the myelination starts at the back and doesn't fully complete up here until you're like in your late teens, early 20s. So like having like a high school kid who's being an idiot and doing like, they know they shouldn't do it and they do it anyway, or like, it's not necessarily just, impulse control because of lack of willpower. Some of it might be the actual like maturing of the circuits of the brain that are actually doing impulse control and decision making. So it's an interesting thing. Like it's always, thinking about the neuroscience of this always is tricky because you still want to hold people accountable. But then you also want to, be aware that, what can I hold someone accountable to? Or maybe there should be you have to be a little careful about what is even possible. It's like you would not get mad at a little toddler for not being able to go jog around the track because their nervous system hasn't actually matured in terms of operating their musculoskeletal system yet. And there's evidence that some of our more subtle executive functioning is not fully developed until you're kind of into young adulthood, which is, again, it's fascinating. Impulse control, planning, executive. And then, like I said, a lot of stuff is what we call bottom up, where kind of information is coming from, well, not from the motor, bottom up, where information is coming up from lower levels of processing to this kind of higher level. But then at the same time, there is a lot of wiring where you have your attention and things like that affecting the operation of the other parts of your brain that are sending the information up in the first place. So it's not all one way from lower to higher. Cerebral cortex continued. Another important point about the cerebral hemispheres, as well as the rest of the brain, is this idea of lateralization. So let me bring that up. The right and left sides of the brain. The most important thing is just the physical anatomical wiring, that in general, the brain is cross-wired with the body. So right side and vice versa. So the right side of the brain is connected to the left hemisphere, and the left side of the brain is connected. What am I? Left side of the body. I didn't mean brain. I am. Right, hold on, let me back up. So when I edit this, I will create a new reality where I never spazzed on you. All right, so we're going to talk about lateralization. And this idea that the opposite sides of the body are connected to the opposite sides of the brain. So the right side of the body is connected to the left hemisphere. And the left side of the body connected to the right hemisphere. You know, this is happening due to an actual crossing over of the axons from one side to the other side of the body. The official word is decussation. This just means crossing over. If you remember from your anatomy, there was the decussation of the pyramids in the brainstem. That's where like most of your motor axons move from right to left or left to right to connect to the other side of the body. Sensory things also cross before they end up in the cortex. This is important when you're worrying about what kind of deficits you're going to see after a stroke. So for instance, let's say somebody has a stroke where they have damage to their right precentral gyrus. What kind of deficits would you expect for if you had a lesion to the right precentral gyrus?
If they had damage to the precentral on the right side, they would have damage to their like motor ability on the left side. Exactly.
So you'd have problems with motor on the left side of your body, probably some kind of paralysis. So, and I should say, in mnemonics, the way when I was learning this, and I had to remember which was sensory and which was motor, I used the alphabetical order of M and S. The motor, M comes before S in the alphabet, and M is in front of S. The motor is in front, the precentral gyrus, the sensory, somatosensory is on the rear side. So that's how I remembered it, is M comes before S, so M is in front of S, motor, somatosensory. So when you're, because you do need to know that. That'll show up on an exam or something. You need to know somatosensory is the postcentral versus precentral is the motor. So lateralization, some of it is just physical connection, both motor and sensory, which side of the body is connected to which side of the brain. Lateralization is also important when we think of language. Yeah, for like 90% of people, the main language is on the left side of the brain. For the rest, 5%, it's on the right. Those people tend to be lefties. The people who have their language centers on the right side of the brain on the left side of the brain tend to be right-handed people. What am I saying? I don't know why I'm spazzing on you guys so much. So most people, language is in the left hemisphere, and these are the typical, most people are right-handed. 5% of the people, it's right hemisphere. These tend to be lefties. And then maybe 5% it's mixed. Right, so this is really important actually though, if you're doing brain surgery on somebody, for instance, you want to know which side of their brain is their primary language. Like if you're going in, if you're going in near the motor cortex, and after surgery, maybe their hand is a little more clumsy than it used to be. It's like, I mean, unless you're a professional musician or something, it's not going to be absolutely debilitating. If your language ability is impaired, that's totally debilitating. So before people do brain surgery, they like to know which side of the brain is the primary language on, so they know whether or not they need to be super careful around these language areas. There's actually this test, I forget the name of it, where they actually put an anesthetic up your carotid arteries, and you can anesthetize one or the other hemisphere selectively. And yeah, my friend actually watched this happen to somebody, and they're there, they're talking, you numb out one hemisphere, they keep talking, you numb out the other hemisphere, though, it's like they go mute. So you can actually relatively non-invasively, if you have to figure out what side is somebody's language on, just put some anesthetic up one or the other thing. So language. I should mention other things about lateralization that is in pop culture and is borne out by studies. The right side, people talk about, the right brain, more creative, left side being more logical. That does seem to play out. Like the processing on the left side tends to be more of the kind of analytical, calculating. The right side, in fact, thinking about language even. When I say language is in the left hemisphere for most people, it's the semantic content of the language. Like, what do the words mean? But a lot of communication in language is not about just the definitions. It's about, nonverbal communication, your expression, the intonation, the tone of voice. So even if the left side of the brain is figuring out what do these words mean and how do I string them together properly, the right side is still getting important information that's more in the affective, emotional domain. Again, classic example I like to do would be, I'm gonna have to pick somebody that's knows that I'm joking. Like I see John there. So I could say, hey, John, it's good to see you in the Zoom meeting. And I could also use the exact same words like, oh, hey, John, good to see you in the Zoom meeting.
You too, buddy.
Right? It's like the words semantically. If you just wrote down the words and asked a computer to define what's happened, it's the exact same thing. But the expression and the tone of voice makes a huge difference. So that seems like thinking about two sides of the brain doing the same thing, but different aspects of the same thing. That's like an example there. They're both working with the language, but kind of different qualities there. Other things about language. The language areas are usually thought of as two main places. The reality is more complicated, but it's a good first approximation. So we're going to talk about language areas in the brain. Again, this is still cortex. We're talking about just in the left hemisphere for most people, or in the right hemisphere for a subset of people. The two areas that you should know, kind of in the frontal area, is kind of connected, is kind of near this motor association, is Broca's area. This is about production of language. This is, again, it makes sense, it's in motor. This is how that magical link happens between my idea, I want to talk, and the words coming out of my mouth, as all this complicated motor programming happens to make my lips and my tongue and everything do their thing. When there is damage to Broca's area, you end up what's called expressive aphasia. So aphasia is the more generic term for problems with language. So if you have damage to Broca's area, you might know what you want to say, but you have trouble making the words come out. That's why it's called expressive aphasia. I can't express myself. It can also be called non-fluent aphasia, because you're not fluent. You can't make words come out easily. The other area is kind of more at this junction of the parietal temporal. It's called Wernicke's area. And again, these names come from these neurologists from the 1800s who had patients that exhibited different kinds of aphasia. And after they died post-mortem, they could see what part of their brain was damaged. Like Broca, Paul Broca had his famous patient, he called him patient Tan, because this person could only go tan, 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 tan. Every time he tried to talk, he'd go tan. And when he died, they were able to see, oh, it was this region in particular where he had suffered damage. Wernicke's area, on the other hand, is about understanding language. So when people have damage here, rather than having trouble making language, you have problem making, either understanding what people are saying or saying words that make any sense. So when people have aphasias here, we say they're receptive aphasia, meaning I'm having trouble receiving your meaning, understanding what you're saying with your words. It can also be called fluent aphasia, because they don't have a problem making the words, but the words end up just being nonsense. Kind of these word salads come out. You can get mixes. Sometimes you have a stroke and an artery that supplies both these regions. So you have some mix of the two. And sometimes it's hard to know clinically if somebody isn't speaking because they can't, or isn't speaking because they don't understand, or they don't know what they want to say. So it can get tricky. But I'll show in your module, I have a couple of nice videos. And there's lots more you can find out there in the world as well. But these are just two that I think are useful if you haven't seen them. Where is it? Here, aphasia examples. I have it. It's just here in the thing for this week, in the module for this week. Here's Broca's non-fluent expressive aphasia. So this is somebody who has damage to Broca's area. Everything else is fine. There's no cognitive deficits or anything. It's just that part of his brain that converts what I want to say to like blah, 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 blah is damaged.
Google Fi, a phone plan by Google. It's easy to switch to Fi from the comfort of...
Can you tell us your name?
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago.
Okay.
And
what did you used to do?
Worked Autodesk, seven sales and worldwide, and very good. Yeah.
Okay. And who are you looking at over there when you turn your head?
That's my wife.
Okay. And why is she helping you to talk?
She's a speech.
So you have trouble with your speech?
Yeah.
And what's that called?
aphasia.
All right.
So you get a sense. I mean, this guy is actually pretty far post like therapy and stuff, kind of working on stuff. There's a girl. Why is this back? Here's another one, just to give you a sense.
Okay. So what's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
You're 19. 19. Yeah. And what happened to you?
Stroke.
You had a stroke last year.
Yes.
And what happened? Can you remember what happened?
School and English class.
Okay.
And I book and I read it aloud. But I can't because stroke.
Yeah, so again, you get an idea. She knows what she wants to say, but she just can't make it happen, which is really different from the other kind of aphasia where your motor production of language is totally intact, but your ability to understand words is off. So like this dude here is an example of fluent aphasia, of where you can see he's gonna chat your ear off, but the more you listen to him, it doesn't make any sense.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment, talk with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him, with luck for him.
So we're on a cruise and we're about to get
We
to
will
June
store it
now.
right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
With the iPad that we were doing. We, like here?
I might change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're all with them. Other people are working with them. I'm very happy with them.
Good.
This girl was fairly good and happy. And I play golf and hit other trees. We play out with the hands.
So you can get a sense like this dude, it's really hard to tell with him what's going on, if he even knows what he's trying to say, or he understands. He probably doesn't even understand the questions, and he's just trying to kind of compensate by being sociable somehow. But
I have a question. So in that second one, it's like you're saying that they can't understand. So you're saying like he can't understand the language that other people are saying to him, as well as
it's probably
how to like put his own like words together to make sense.
I think both of those. I think, and there's different severities and different versions, and some versions affect more just the spoken versus spoken and written, and so there's lots of different subtypes. But I think it's a mix of not being able to understand what the words that you're hearing mean, as well as being able to string together meaningful words as well. So, yeah, so you can, yeah, it's not uncommon to have aphasia, especially after a stroke. So it's, and again, that plasticity, often when people have strokes, there's, some recovery after for a while. What else
Do some forms of aphasia happen from like seizures and epilepsy as well? maybe not as like permanent, but
I
I had a
think,
friend who has epilepsy and he had a lot of trouble talking after his episodes.
yeah, so I mean, depending where that electrical storm was, if it was affecting those speech areas, I would imagine that would be the case. So yeah, no, that's not surprising. So language aphasias, again, mainly on the left side for most people, but sometimes on the right. What else do I want to say? Last little bit is, I've been drawing kind of the brain just from the side like this. If we do like a cut down the middle, so we're actually seeing the two halves of the brain, the two hemispheres. So this is the cortex, but then this is that little called the longitudinal fissure, the kind of groove between the two halves of the walnut. Here's the corpus callosum connecting the two hemispheres. This little gyrus right here is an interesting one. It's called the cingulate gyrus. I want you to know it because it's going to be coming up a little later. Let me see if I have a picture I can show you. Yeah, here, let me show you on the PowerPoint. No. So if we look here at the brain that we've been looking at all along, and again, this is like the brain from the outside. This is looking at that inside between the two different hemispheres. And right here is that cingulate gyrus. It's kind of in the midline and just above the corpus callosum, but it's a ridge of gray matter. So it's nothing beyond just being a ridge of gray matter. It's involved in emotional processing, and it'll come up when we talk about the limbic system. So you should know about it there. It gets implicated in balancing pragmatic versus emotional qualities of decision making. People study decisions like, if I the classic thing is there's a train about to go off into a ravine because the bridge is out. And if I push the switch on the track, the train will go off on a siding, but there's a person on the siding, so someone's going to get killed when the train goes off on the siding. But at least only one person died instead of everybody on the train who died if the train would have gone into the ravine. And most people in that case do the pragmatic calculation. Well, one person died versus like the train full of people died. So I guess I would flip the switch. But if you reframe it so it's got a much ickier emotional component, like you have to, take somebody and jam them under the wheels or something to stop the train. So it's still like pragmatically, the math is the same. Like you do something that either kills one person or does the whole train dies. People are much more likely like, I can't do that. I can't just take this person and shove them under the track. So I'm just going to step back and it's going to be horrifying. But, so that kind of thing. You can do all sorts of studies showing that we're pretty irrational in terms of our decision making. It's like this balance between pragmatic analysis and the emotional feel of things, and where we land seems to depend, at least in part, on cingulate gyrus, particularly like the anterior part. It's there's a lot of business people who've been doing more neuroscience, like neuroeconomics, trying to figure out what kind of things cause us to make different choices, and often kind of irrational choices. And often the choices we make are different, whether or not we feel someone's watching us or not as well, which it's not like surprising, but is kind of interesting. John, I saw your hand up.
Yeah, I was wondering, the cingulate gyrus, is that, okay, going back to the train, would the part of you that says, well, clearly let's think of the greater good, would that be the prefrontal cortex, and that's working against or kind of balancing against the cingulate gyrus?
So
the part that says, no, we can't even
the
kill one.
I would say the details I can't say. I mean, that's a good as any hypothesis. When we get to the limbic system and emotions, what we're going to find is we can talk about regions that are involved in emotions. We know regions that get damaged that cause dysregulation of emotions, but exactly what emotions look like and how they manifest in the reality of the circuits, it's still not that clear. So, but yeah, I think that's a good hypothesis. So cingulate gyrus, another area that is involved in emotional stuff is the insula. So this is again a picture of the brain, and here's a cross section, and again you can see the two hemispheres. And here you can see something else though that you couldn't, I haven't really talked about. Where's my little annotator? Like right here, this is this groove, this lateral sulcus where the temporal lobe meets the parietal and frontal lobes here. And if you look, there's actually a little crevasse, and there's this little bit of cortex that's down deep in that crevasse. That is called the insula. It means little island. The insula has regions that give you sense of kind of your proprioception, what your body is up to. You know, so, is also very implicated with emotions as well. So it will come up when we talk about the limbic system, right? I mean, a lot of times, you know, depending, some emotions get really complicated, like, you know, but some emotions, even back in the days of what's his name? The guy who wrote the varieties of religious experience. Oh, I'll remember his name. There was a psychologist like that was pretty amazing. He's the guy who came up with the idea of stream of consciousness and stuff. But he would talk about emotions being more something that happened in your body first, and then your brain kind of puts a name on it. Like, you know, your heart's speeding up and you're getting sweaty, and it's like, I must be kind of scared and nervous here. But there was kind of a physiological response, and the emotion was something that is your subjective experience of your body having some reaction, not so much a mental construct that then results in some effect on your body. And, you know, again, that's even the idea of feelings, the word we use, you know, my feelings, like feel is like touching shit stuff. You know, it's kind of interesting when you think about it. So we will see the insula a little later when we just talk about regions that are implicated in processing of emotion. And it's also involved in just processing basic readout of the status of your body as well. All right. Time is it? 10:46. Trying to think if there's anything that I want to say before we move on. Okay, just to reiterate, everything we've been talking about, all of these sensory cortices, the prefrontal areas, this is all cortex. It's all happening on this thin layer right at the surface. You know, it's actually a pretty large surface area. It just happens to be all crumpled up into your skull and getting all wrinkled. That's why you have all the sulci and gyri, the sulcus, the grooves, the gyri, the ridges. On Tuesday, we'll finish the cerebrum by looking at some of these deeper gray matters, the lentiform nucleus, the caudate nucleus, things like that, which are going to be involved in some motor stuff as well as some emotional stuff. So we're not done with the cerebrum, but we've done a lot of it. We've done the kind of core of the cortex, which is where a lot of the interesting stuff is happening. Okie dokie. Cortex.